Make me a host. I'm not able to share the screen. So in the previous classes, we were learning about the structure of glucose. Now we will learn the ring structure disaccharides so that the carbohydrates can be finished today, and then we can go for other biomolecules. Now 6 carbon atom in a straight chain. Carbon atom aldehyde. CHO is an aldehyde. And the second carbon atom, OH on the right-hand side. Fourth carbon atom, OH on the right-hand side. Fifth carbon atom, OH on the right-hand side. The sixth carbon atom is a primary alcohol, CH2OH. Now, if OH is on the right-hand side, H is on the right-hand side, OH on the left-hand side. This is how we remember the structure of glucose. Now, you write the structure of glucose in another form. Instead of showing the carbon-oxygen double bond on the right-hand side, we will show it in the left-hand side. The same structure I am writing. The same structure I'm writing. This is also glucose. This is also glucose. This is the open chain structure of glucose. This is the first carbon atom, there's a fifth carbon atom, and this is the sixth. Similarly, here the first, fifth, and sixth. In order to write the ring structure, now the hydrogen atom of the fifth carbon atom, gets bonded to oxygen of the first carbon atom. Meaning, here this hydrogen, we had OH. From this, the hydrogen atoms attack oxygen. So therefore, the valency of carbon oxygen increases, therefore pi bond breaks. When pi bond breaks, now the valency of carbon is also not satisfied. There are only 3 bonds. Therefore, the first carbon atom and the fifth carbon atom will get bonded through oxygen. This is a ring structure. Similarly, now if we go for this, the hydrogen atom of the OH group of the fifth carbon atom gets bonded to oxygen atom of the first carbon atom. Therefore, the pi bond breaks. Now, the first carbon atom and the fifth carbon atom are bonded through oxygen. So these are the ring structures of glucose. Now, this ring structure, you can see here in this will be called as alpha D plus glucose. This will be called as alpha D plus glucose, and this will be called as beta D plus glucose. Now, the configuration is different only at the first carbon atoms. Here, the first carbon atom OH is on the right-hand side. Here, the OH is on the left-hand side on the first carbon atom. Since the configuration differs only at the first carbon atom, this alpha D glucose and beta D glucose are called as anomers. They are called as anomers. Anomers are compounds having the same structure, only at the first carbon atom, the hydrogen and the OH groups are differently arranged in space, or spatially they have different arrangement only at the first carbon atom. Therefore, they are called as anomers. Now, this alpha carbon atom and beta carbon atom will exist in equilibrium with the open chain, structure. We have learned the open chain structure in the previous class. I told you to remember, and today also I started with the open chain structure. Now, we'll have to learn to write the Haworth structure. Here, the plus sign shows that both the anomers are dextrorotatory. This plus sign indicates both the anomers, both the molecules rotate the plane polarized light clockwise. Therefore, they are called as dextrorotatory. Dextrorotatory. Now, coming to the Haworth structure. Now, what is the Haworth structure of glucose? In order to write the Haworth structure, this Haworth structure is also called as a ring structure. Is also called as a ring structure. Now, in case of Haworth structure or the ring structure of glucose, we call this Haworth structure of glucose as a pyranose ring structure. Why we call it as a pyranose ring structure is because now pyron is a 6-membered ring. Since glucose contains 6 carbon atoms, it will have a 6-membered ring. Therefore, it is called as pyranose. OSE stands for sugar, I told you, or glucose with sweetness, sugar. Now, coming to the Haworth structure, I will write the Haworth structure here so that you can see this structure and write. Now, in order to write alpha D plus glucose, first we write a hexagon like this. We write a hexagon like this. Now, here we write an oxygen atom, and we number this as 1, 2, 3, 4, 5. The first carbon atom is the 5th carbon atom. The first and the 5th carbon atom is bonded through oxygen. You can see the first and the 5th carbon atom is bonded through oxygen. Now, in order, and you can write this, all are in one plane. The 5 carbon atoms and 1 oxygen, they are in one plane. So we'll write a bond like this. One is above the plane, below the plane, above the plane, below the plane, above the plane, below the plane. Now, we'll have to, in order to show this H and OH groups, now whatever is on the right-hand side, you will write it in the lower plane bond. So the first carbon atom, OH, is on the right-hand side. So OH and H. The second carbon atom also, OH and H. The third carbon atom, you can see hydrogen also on the right-hand side, hydrogen and OH. The fourth carbon atom, this is 4th carbon atom, OH on the right-hand side. So it will be OH and H. And on the 5th carbon atom, if we take this as a 5th carbon, this is left and this on the right-hand side. Since this is on the right-hand side, it has to be written on the lower bond. But due to steric hindrance, the configuration of the 5th carbon atom flips. The CH2 group goes on the above and the hydrogen comes in the below. This is alpha D plus glucopyranose. Pyranose means a 5-membered ring, a sugar which has a 6-membered ring, not 5-membered ring, 6-membered ring. Now, likewise, if we take the beta D glucose, the Haworth structure, again, we will write a hexagon. And here we'll write an oxygen. Number this as 1, 2, 3, 4, and 5. And the procedure is same. Whatever is on the right-hand side, on the on the lower plane, and whatever is on the left-hand side on the upper plane. So here it will be H and OH. Here it will be OH, H, H, OH, OH, H, and here it will be CH2OH and H. Only at the first carbon atom in alpha D plus glucopyranose, hydrogens on the upper plane, OH on the left hand side, LW the lower plane. Here in beta D glucopyranose, I'm not able to, this is beta D plus glucopyranose. Here the OH is in the upper plane and H is in the lower plane. This is the structure of glucose. This is the Haworth structure of glucose. Now, coming to fructose, we will learn about the Haworth structure of fructose. Now, before we go for the Haworth structure of fructose and other molecules, we should know what is mutarotation. Now, mutarotation, 2 forms, alpha and beta glucose, exist in separate crystalline form and have different melting points and optical rotations. So alpha D plus glucose and beta D plus glucose exist in different or separate crystalline forms. and have different melting points and optical rotations. Alpha D glucose has specific rotation of 111 degrees, and beta D glucose has specific rotation of plus 19.2 degrees. When alpha form is dissolved in water, its specific rotation falls until a constant of 52.5 degrees is reached. When beta D plus is dissolved in water, the specific rotation increases and becomes constant at 52.5 degrees. This spontaneous change in specific rotation of an optically active compound with time to an equilibrium is called as muta rotation. Now, coming to the next monomer, that is fructose. Now, fructose, we have, I'll write fructose here because there is no space. I'll write the structure of fructose here. Now, fructose, we write 6 carbon atom in a straight chain. The first carbon atom and the 6th carbon atom, we write a primary alcohol. And the second carbon atom, a ketone group. Third carbon atom, hydrogen on the right-hand side. Fourth carbon atom, OH. Fifth carbon atom, OH on the right-hand side. So the rest, if OH is on the right-hand side, H is on the left-hand side. This is the structure of fructose. This is the structure of fructose. Fructose is an example for ketohexose. Now, in order to write the ring structure, we can write the ring structure of fructose in this way. I'll write it, and instead of showing the CH2 group OH here, I will write CH2OH on the left-hand side. For one more molecule, I will write CH2 C double bond O and CH2OH on the right-hand side edge. Now here it will be CH, OH, COH, H, COH, H, and CH2OH. This is the open chain structure of fructose. Now this fructose is called as levolose. It's also called as levolose, L-A-E-V-U-L-O-S-E. It is levorotatory. It will rotate the plane polarized light to the left-hand side. Therefore, it is called as levolose. Now, fructose also exhibits in 2 forms, ring structure. Now, the first ring structure, what we're going to learn will be alpha D minus fructose. Now, in case of alpha D minus fructose, so what happens is the hydrogen atom of the OH group of the 5th carbon atom, the hydrogen atom of the OH of the 5th carbon atom attacks oxygen of the 2nd carbon atom. Therefore, what happens is the pi bond breaks. Now, the 2nd carbon atom and the 5th carbon atom are bonded through oxygen. Similarly, here also, the OH group here, the hydrogen atom of the OH group attacks oxygen. So therefore, the pi bond breaks. When the pi bond breaks, now the 2nd carbon atom and the 5th carbon atom gets bonded through oxygen. This is beta D minus fructose. This is alpha D minus fructose. And this is beta D minus fructose. Now, this minus sign indicates it rotates the plane polarized light to the left-hand side, levorotatory. It is called as fruit sugar. Now, this fructose in the Harvard structure, it will have a 5-membered ring. Since it has a 5-membered ring, it will be called as furanose. It will be called as furanose. Now, in order to write the 5-membered ring of alpha D minus fructose, write a pentagon like this. And write oxygen here. Now we can number this as 2, 3, 4, 5. The 2nd and the 5th carbon atom is bonded through oxygen. Now we can write bonds like this. Now OH is on the right-hand side of the 2nd carbon atom, so lower plane, CH2OH. Here it is H and here it is OH. Here it is OH and here it is H. This is on the right-hand side, so it will be CH2OH and H. This will be called as alpha D minus fructofuranose. It's a five-membered ring. Furan is a five-membered ring. So it's a fructofuranose. The next one is beta D minus fructose. In order to understand this beta D minus fructose, we will write again a five-membered ring like this. An oxygen atom here. This is 2, 3, 4, and 5. We'll write the bonds here. Now, CH2OH on the right-hand side. So it comes to lower plane and OH here. H, OH, OH, H. Here it is CH2, OH, and H. So this will be called as beta D minus fructofuranose. This will be called as beta D minus fructofuranose. So these are the Hayward structure of fructose. Now we need to write the Hayward structure of disaccharides. We will write the Hayward structures of disaccharides in a few minutes' time, but remember to know the structures, Hayward structure of glucose and fructose. What we've learned: alpha D plus glucopyranose, beta D plus glucopyranose, and alpha D plus fructofuranose, beta D minus, sorry, this is minus fructofuranose. Based on this, we can write the structures of some of the disaccharides. Now, what are disaccharides? Di stands for two. Saccharides stands for sugars. So carbohydrates, which on hydrolysis gives two or different monosaccharide units, are called as disaccharides. Di stands for two. A disaccharide is a carbohydrate which contains two monosaccharide units. These two monosaccharide units can be same or different. Now, sucrose, you can see sucrose, lactose, and maltose. All these three have the same molecular formula. When we hydrolyze this, they all give two units. One is glucose unit here, and the other one is fructose. So when sucrose is hydrolyzed, we get a glucose unit as well as a fructose unit. When lactose is hydrolyzed, one we will get a glucose unit, the other one we get galactose. So what are the monosaccharides in lactose? The monosaccharide in lactose is glucose and galactose. When maltose, milk sugar, when this is hydrolyzed, we get 2 units of glucose. So we need to learn the Hayward structures of these disaccharides, sucrose, lactose, and maltose. It will be asked for you to write a Hayward structure for 2 marks, possibilities. Now the first one is sucrose. The sucrose is also called as cane sugar because we obtain sucrose from sugar cane. It is also called as cane sugar. It is widely distributed in nature in juices, seeds, fruits, and also in the flowers of many plants. The main source of sucrose are sugarcane juice, which contains around 50 to 20% sucrose. So sugarcane juice contains 50 to 20% sucrose. And sugar beets, which is about 10 to 17% sucrose. So beetroot, sugar beets, when we say beetroot, the vegetable, it contains around 10 to 17% sucrose. Now, the sucrose is colorless, it's crystalline, sweet substance, soluble in water. The aqueous solution is dextrorotatory. Sucrose is dextrorotatory. Sucrose will rotate the plane polarized light clockwise. Therefore, it is called as dextrorotatory. Now, sucrose on hydrolysis gives 2 monosaccharide units. You should remember it is glucose and fructose. Now, widely sucrose is obtained from sugarcane juice, or 15 to 20%, and in beetroot or sugar beets also contains around 10 to 17% sucrose. It is colorless, it is sweet to taste, and it's soluble in water. Now, sucrose on hydrolysis in the presence of an enzyme, invertase, or dilute acids, gives dextrorotatory glucose and levorotatory fructose. So, we learned sucrose, on hydrolysis gives glucose and fructose. Now here they clearly say it is alpha D plus glucopyranose. And here it is beta, because levorotatory, beta D minus fructofuranose. So these are the specific, more apt or appropriate definition for the monosaccharides of sucrose on hydrolysis. Now, sucrose solution, how do we prepare sucrose? Now, this is a mixture. You can see here hydrolysis, fructofuranose. Since levorotation of fructose is minus 94.4 degrees, is more than dextrorotatory of glucose. Glucose is 52.5 degrees. The mixture is levorotatory. The mixture of glucose and fructose is levorotatory because levorrotation of fructose is more, minus 52.4 degrees. Thus, hydrolysis of sucrose brings a change in the sign of rotation from dextro to levo. So sucrose solid as such, it is a dextrorotatory, but in solution, it changes from dextro to levo. And such change is known as inversion, and the mixture is also known as invert sugar. Sucrose solution on fermentation by yeast, enzyme invertase converts it into glucose and fructose. This is not glucose. This is not glucose. This is sucrose. C12H22O11 is sucrose. Sucrose on hydrolysis in the presence of enzyme invertase, it brings hydrolysis fermentation to form one of the molecule glucose, the other one is fructose. The enzyme zymase converts glucose and fructose into ethanol, giving carbon dioxide and releasing energy. So carbohydrates are source of energy. Now, if you can remember the Harvard structure of glucose, alpha D plus glucose, I taught you today to write a 6-membered ring. It's a glucopyranose rings. So I told you to number this carbon atom as 1, 2, 3, 4, and 5. Now, since it's alpha D plus glucose, hydrogen atom will be on the right above plane, OH here, H here, OH here. Here it will be OH, H. Here it will be OH, H, and CH2OH here and H. This is alpha D plus glucopyranose. This is alpha D plus glucopyranose. Now, beta D minus fructofuranose. In order to understand beta D minus fructofuranose, I told you to write a 5-membered ring and an oxygen atom here. I told you to number this as 3, 4, 5, because in fructose, the second and the 5th carbon atom is bonded through oxygen. And we told you to write bonds like this. And here we have CH2OH. Here it is OH. Here it is OH, H, H, OH, H, OH, CH2OH. This is beta D gluco, beta D minus fructofuranose. Now, in order to write the Harvard structure of sucrose, now I try to elongate this OH bond here and this OH bond here. I try to elongate. This is an OH bond here, and this is also an OH bond. I elongate. When these two OH bonds come very close up, it should be drawn in a straight line. You can draw it in this form also, not a problem. So water gets eliminated. When water gets eliminated, this water gets eliminated. When water gets eliminated, the first carbon atom and the first carbon atom of glucose and the second carbon atom of fructose will get bonded between each other through oxygen. This kind of bond, carbon 1, oxygen carbon 2, is called as glycosidic linkage. Glycosidic linkage. Now, this glycosidic linkage is a bond between carbon and carbon through oxygen of different monosaccharide units, not between the same monosaccharide. See, carbon 1 and carbon 5, this is not called as a glycosidic linkage. So carbon of one monosaccharide units, carbon of the second monosaccharide unit, when they are bonded through oxygen, then we call those kind of bonding to be a glycosidic linkage. In case of sucrose, the glycosidic linkage is between first carbon atom of glucose unit and the second carbon atom of fructose unit. This is the Hayward structure of sucrose. You should remember the ring structure, they will not ask. Now coming to maltose. Maltose is also called as malt sugar. It is present in germinated seeds. It is prepared by partial hydrolysis of starch, either with dilute mineral acid or with enzyme diastase. So maltose on hydrolysis with the mineral acid like HCl, H2SO4, a dilute, or with the enzyme diastase, we get two monosaccharide units. It is dextrorotatory in nature. So maltose, diastase stage is written. Diastase, it is TASE. Now diastase is an enzyme which hydrolyzes the maltose into glucose units. So maltose on hydrolysis gives two glucose units. Maltose on hydrolysis gives two glucose units. This is maltose. We'll write a specific molecular formula of maltose, that is C6H12O6. This on hydrolysis in the presence of the enzyme diastase, or maybe the dilute acid, we get 2 units of C6H12O6, 2 units of glucose. Now 2 units of glucose, we can represent it like this. Now this is glucose. I'm just making a change here, then we will. Here we have OH group. Here also we have OH group. Here also we have OH group. See, this at first carbon atom, hydrogen on the upper plane, OH on the lower plane. The same thing here also. It is alpha D plus glucose units. Both are alpha D plus glucose units. Under the fourth carbon atom, you can see here hydrogen on the upper plane and OH on the lower plane. When these two OH are close, what happens is water gets eliminated. When water gets eliminated, we have this oxygen. This oxygen will be bonded between first carbon atom, one glucose unit, and fourth carbon atom, the other glucose unit. So we get a glycosidic linkage. This OH is not to be mentioned, it is wrongly printed. So first carbon, C1 of one glucose unit and C4 of the second glucose unit. So in case of maltose, the linkage, the glycosidic linkage is between first carbon atom of one glucose unit and the fourth carbon atom of the second glucose unit. Maltose is an example for reducing sugar. It reduces tolerance reagent and Fehling's solution. Sucrose is an example for non-reducing sugar. Remember, sucrose is an example for non-reducing sugar. Maltose is an example for reducing sugar. The next we have is lactose. This lactose also has the molecular formula C12H22O11. This on hydrolysis. This maltose is called as milk sugar. It's called as milk sugar. This on hydrolysis in the presence of an enzyme emulsin or in the presence of an acid. This also gives you one glucose unit and one galactose unit. The molecular formula of glucose and galactose are same. So the Hayward structure we'll have to write for maltose. The Hayward structure is this. The Hayward structure is this. This is galactose and this is. The structure looks similar for galactose and maltose. The only difference is the fourth carbon atom. The fourth carbon atom, here the OH is on the upper bond for galactose. Here the OH will be at the lower bond. We will draw the structure properly. This is not apt. It's not clearly mentioned. Anyways, we will draw it again. This is galactose. Sorry, I will write it neatly in some other place, a larger space if I have. Yeah, I will write here the structure of galactose. So we will write the structure of galactose, a pyron ring. So it's carbon 1 and 5. So we will write this as OH, and this is H, and this is H, and this is OH, and this is OH, and this is H. This is OH, and this is H, and this is H, and this is CH2OH. This is galactose. Now, in case of glucose, again a pentagon, a pyron ring. Here it will be OH, H. This is H, OH, H, OH. Here it will be OH, H, and CH2OH. And this is glucose. This is glucose. This is galactose. Now, if you observe very carefully, the structure looks similar. Only at the fourth carbon atom you can see the configuration is different in case of glucose and galactose. Now, what happens is when these OH groups come closer, I'll show the OH group like this here. This OH group, I bring it here. This OH group, I bring it here close. Now, what happens? The bond OH groups are it's close to each other, therefore the water gets eliminated. When water gets eliminated, now the first carbon atom of galactose and the fourth carbon atom of glucose are bonded to oxygen. So this is C1OC4. This is again a glycosidic linkage, C1OC4. But this is between galactose and glucose. This is also an example for reducing sugar. Lactose is also an example for reducing sugar. It reduces tolerance reagent and Felling's solution because it breaks and free aldehyde groups are released at carbon 1. Therefore, it is a reducing sugar. Now, coming to the last part of the carbohydrate, that is polysaccharide. Poly stands for many. We know that we have different types of different examples for polysaccharide. The first example, what we're going to learn, is called as starch. Starch is also called as amylin. The general formula of starch is C6H10O5 N times. C6H10O5 N times. It's a polysaccharide, storage polysaccharide in plants. The main source of starch are wheat, maize, rice, potatoes, barley, sorghum. It occurs in the form of granules, which varies in size and shape depending on the plant source. Starch is a white amorphous powder. It is not sweet to taste. It is not soluble in water. It gives blue color with iodine. Starch can be used as an indicator in titrations. The blue color appears on heating and reappears on cooling. Starch on hydrolysis gives hydrolysis in the presence of an enzyme diastase or dilute acids. First gives maltose and finally D-glucose. So sucrose on hydrolysis in the presence of enzyme diastase, we get maltose. This maltose further undergoes hydrolysis to give glucose units. So the final product on hydrolysis of starch is glucose. Now the starch contains two parts. Starch contains two parts. The first part of the starch is called as amylose. The first part of the starch is called as amylose. It is around 15 to 20%. The second component in starch is amylopectin. This amylopectin is around 80 to 85%. They can ask you to write the partial structure of starch, amylose or amylopectin. So structure of amylose. The structure of amylose. This is the structure of amylose. It is a linear polysaccharide containing alpha D plus glucopyrones. So amylose contain alpha D plus glucopyrones. glucopyranose. We have learned what is glucopyranose, a six-membered ring. And you can see these are glucopyranose units. And the bonding glycosidic linkage is between first carbon atom of one glucose unit with fourth carbon atom of the other glucose unit. So this is easy. It is a structure of amylose. It's a partial structure of amylose. Now, amylopectin is a branched chain polymer. Now, amylose is a water-soluble polymer. It is a linear polymer. It is formed by the linkage of alpha-D glucose pyranose at 1 and 4 glycosidic linkage. Amylopectin is branched chain polysaccharide, insoluble in water and does not give blue color with iodine. It's made up of 15 to 30 D glucose units and joined by alpha-D glycosidic linkage between carbon 1 of one glucose unit and carbon 4 of the next glucose unit. These chains are connected with each other by 1,6 linkage. This is amylose. This is also amylose. But you can see one of the amylose, the OH group, is bonded to the alcohol group of the other linear chain, the CH2OH. This OH and this OH come. Now here we have OH and here it is CH2OH. It's alcohol group here. These OH come closer. Therefore, this water gets eliminated, H and OH, and a link is formed. So this glycosidic link is between carbon 1, oxygen, and carbon 6. This is the sixth carbon atom. These are the sixth carbon atom. So this is the fifth carbon atom, this is the sixth carbon atom, C6. So it's called as 1,6 linkage, glycosidic linkage. This is the partial structure of amylopectin. This is amylose unit, this is amylose unit. These are connected with each other through 1,6 glycosidic linkage. Next, cellulose. Cellulose also has the general formula C6H10O5 N types. We do not have to learn more about the cellulose. It is a cell wall. It's a major constituent of cell wall of plants. The major constituent of cell walls of plant is cellulose. Cotton is a pure cellulose. Cotton is a pure cellulose. Next polysaccharide we have is glycogen. We're supposed to learn. Glycogen is the polysaccharide. Glycogen can be asked for a few marks. Glycogen, it is a storage polysaccharide. Glycogen also has the molecular formula C6H10O5 N times. C6H10O5 N type. It's a storage polysaccharide in animals. It's a storage polysaccharide in animals. Excess of glucose in animal bodies is stored as glycogen in the liver. Excess glucose in animals, human bodies, they are stored in the form of glycogen in the liver, muscle, and brain. Its structure is similar to starch, amylopectin. Its structure is similar to starch, amylopectin. So it is also called as animal starch. Glycogen is also called as animal starch. During starvation, during fasting, during starvation, glycogen is broken down to glucose in the presence of enzyme, and glucose supplies energy. Thus, glycogen meets immediate energy demand of the animal body. So this ends carbohydrate. The second biomolecule, what we're supposed to learn, is called as proteins. Proteins are high molecular mass compounds or high molecular mass complex biopolymers of amino acids. Proteins are biopolymers. They play an important role in development and maintenance of life process. They are of prime importance in plant and animal body. The protoplasm of plant or animal cell contains 10 to 20% protein. The protoplasm of plants or animal cell contains around 10 to 20% proteins. All proteins contain the element carbon, oxygen, nitrogen, and sulfur. Sulfur is not present in all, but all proteins, contain sulfur. Some of these may also contain phosphorus, iodine, and traces of metals like iron, copper, zinc, manganese. All proteins on partial hydrolysis gives peptides. which on complete hydrolysis gives amino acids. Now, what is amino acids? Amino acids. Amino group is NH2. Acid, when we say it is COOH. A molecule which contains amino group and acid group together is called as amino acids. Now, for an example here, you can see this molecule contains amino group as well as the acid group. This is the general formula of amino acid. But in case of proteins, all the amino acids are alpha amino acids. What do you mean by alpha amino acids? Now, we have an acid group as well as an NH2. As per the IUPAC nomenclature, acid is given the preference. This becomes a primary functional group. This becomes a substituent. Now, carbon atom attached to the function group will be called as alpha carbon atom. So this carbon becomes alpha carbon atom. So amino group is bonded to alpha carbon atom. Therefore, this will be called as alpha amino acids. So all the proteins are made up of alpha amino acid. The general formula of alpha amino acid is R, CH, COH, and NH2. The NH2 group is present in alpha position. Now, if I write an example like CH3, CH, NH2, and COOH, this is an example for alpha amino acid because the amino group is present at alpha carbon atom. If I write an example like the CH3, CH, NH2, CH2, and CH2 and COOH. This is alpha carbon atom, this is beta. At beta position, we have NH2. So this is beta amino acid. So proteins do not contain beta, gamma, or delta. So proteins are made up of only alpha amino acid. So therefore, the general formula of alpha amino acid is this: RCH, NH2, COOH. So we can say alpha amino acids are the building blocks of proteins. Or we can simply say proteins on hydrolysis gives alpha amino acids. The statement is mentioned here, proteins on hydrolysis and gives alpha amino acid. So this is the general representation of alpha amino acid. Now, alpha amino acids are classified into based on their properties. Now, based on the properties, when we say I'm looking out for space, now classification of amino acids. Classification of amino acids, alpha amino acids. Now, the first classification is based on the nature. The first one is based on the nature. Now, under that, first will be called as acidic amino acid. Now, acidic amino acids. Now, if for an example, if I write CH2, COOH, CH, NH2, COOH, this is an alpha amino acid, no doubt about it. This is alpha carbon atom. Now, you can see we have two COOH groups and one NH2. Therefore, this amino acid will be acidic in nature. So, example for acidic amino acid is aspartic acid. Aspartic acid is an example for acidic amino acid because we have two COOH group and one NH2 group. NH2 is basic in nature. Now, the next is basic amino acid. Basic alpha amino acid. When we say basic alpha amino acid, if I write the basic alpha amino acid, an example like this. This is amino acid, no doubt about it. I'll tell you how to remember the structures a little later. This is lysine. It's an example for basic amino acid. They'll ask you, give an example for a basic amino acid. Lysine is an example for basic amino acid. We can see here we have two NH2 groups and only one OH. Therefore, the nature of this will be basic. The third amino acid based on the nature is neutral amino acid. This neutral amino acid, we can write an example like this: H, CH, NH2 and COH. One example, I mean, COH, one COH and one NH2. Therefore, the pH will be around 7. It will become a neutral amino acid. It will be neutral amino acid. So based on the nature, amino acids are classified into acidic, basic, and neutral. Now, based on the requirement, these amino acids are classified into essential and non-essential amino acids. Now, what are essential amino acids? What are essential amino acids? Amino acids which are required for the body, which are supplied through diet, will be called as essential amino acids. So remember, we have two types of amino acids based on the requirement. One is essential amino acid. The other one is non-essential amino acids. Now, what is this essential amino acids? Amino acids which are not synthesized in the body. Amino acids which has to be provided or given through food is called as essential amino acids. So essential amino acids are not synthesized in the body. Essential amino acids are not produced in the body. They have to be supplied through diet or through food. Those are called as essential amino acids. Example for essential amino acids important: arginine, methionine. These are all essential amino acids. So amino acids which are produced in the body, amino acids which are synthesized in the body, which is not required for us to take through diet, they are called as non-essential amino acids. There's non-essential amino acids. Example, glycine is an example for non-essential amino acid. Glycine will be an example for non-essential. All right. Glycine is an example for non-essential amino acid. Alanine is an example for non-essential amino acids. So amino acids which are not synthesized in the body, amino acids which has to be supplied through diet, will be called as amino acids. Example, arginine and methionine. Non-essential amino acids which are synthesized in the body, which are not necessary for us to be supplied through diet, will be called as non-essential amino acids. Example for non-essential amino acids, glycine and alanine. So non-essential amino acids are amino acids which are synthesized in the body. They're produced in the body, so need not to be taken through diet. Those are called as non-essential amino acid. The further classification and the properties of amino acid we will learn tomorrow or